So just to start with, just a bit of context around who I am and what I do. So I work with Agriculture Victoria. I'm based in Horsham, and I work within grain quality assessment. So our lab group, well, much of what we do is in supporting the release of new grain cultivars by assessing quality traits of grains and then providing that information to breeders so that they can make selections based on improved quality traits. We also develop capability in grain quality assessment methods, and my role specifically is in developing new technology for assessing quality traits of pulse grains. So the context of my project is in the visual appeal of pulse grains. I'm interested in the colour, size, shape, as well as defects and harvest damage, contamination in samples, that sort of thing. But a lot of those traits are really subjective to measure. So you might be familiar with the process of comparing a grain sample to standard colour charts. Different people will have a different interpretation of what's sound grain and what's defective. So my project tries to remove a lot of that subjectivity from the assessment by using image analysis. So the broad aim of my project is to develop rapid and objective screening methods which accurately quantify visual traits of pulse grains. Oops, sorry. Is the video playing? Yeah, try again. Oh, there we go. So this is the instrument that we're using for collecting images of pulse grains. It's high throughput, so a sample of about 1,500 seeds will travel through that. It can be analysed, like images collected and then analysed for quality in less than 2 minutes. So you can see that it picks images of individual grains as they travel through. So what happens is the sample goes into this hopper, it's dispersed onto a conveyor belt. Conveyor belt carries it through into a camera unit in there. It's imaged under 6 different wavelengths of light, and it's also scanned by laser for surface height. So we get, for every individual seed, we get 6 colour intensity images, and we get a 3-dimensional image from the laser as well. And that gives us data on how surface height and colour is varying across the surface of the seed. And from that we can look at things like dimpling in the seed, we can look at the size and shape, you can look at defects, and how colour varies across the seed, or wrinkling in the seed coat as well with chickpeas. So image collection is the first step, but what I spend most of my time on are these 2 middle steps. So first of all, image processing, that's getting features out of the image, looking pixel by pixel through the images to find like how or patterns, I guess, in the images, in the pixel values, and dimensions of the seed and whatnot. And then the next step is relating that to actual physical traits of the grain, so using mathematical modelling to relate all of that pixel information to physical data. And the overall aim then is to get this assessment of pulse grain quality from the colour to size, shape, and just overall visual quality of the grain. So, overall, I think that was the same one, sorry. So we see the main applications of these algorithms as being in these 3 areas. So plant breeding, first of all, in supporting the release of new cultivars, so providing a method by which traits can be assessed objectively. In the pulse industry as well, providing tools for objective and consistent assessment of visual quality of the grain. And in research agronomy as well, to provide methods to quantitate the different responses from different pulses under the various agronomic treatments. So just to finish off, I've got a couple of slides of results so far and what we're seeing with the image analysis. This is seed size distribution of 4 different pulse types. So we've got lentil, field pea, chickpea and faba bean. On the x-axis there is the size grade, or your seed diameter, if you will. And then the charcoal bars are seed size distribution measured through sieving, the standard sieving method. And golden bars are the predictions from image analysis. So you can see that it's predicting quite well. The shape of the distributions are very similar. Some of the variability in it is actually variability due to the sieving method itself. So, for example, chickpeas don't sieve very well because of the shape of the grain. So that won't, the image analysis method doesn't always match up perfectly because sometimes the beak will get stuck in the sieve. So this is for chickpeas. So smaller chickpeas will often get stuck in a larger size group than what they should. A few more results. On the right-hand side, lentil seed size index predictions. So on the x-axis, you've got seed size index measured through standard sieving method. On the y-axis, you've got the image analysis predicted seed size index. The grey straight line there is the one-to-one correspondence, so closer the data points are to that line, the more accurate the prediction is. And then on the left-hand side is prediction of 100 grain weight for various field pea samples. On the x-axis is the manual predictions, so counting seeds out and weighing them off. And then on the y-axis is the image analysis prediction of 100 grain weight and one-to-one correspondence as well in that graph. So you can see that, the image analysis methods are matching up really well with the standard methods for measuring size. So that's, I guess, an overview of where we're up to at the moment. And where we're heading from here is looking more at colour traits of the grain and contamination in samples, defect and assessment as well, and looking at milling efficiency and that sort of thing. So traits that are important for marketability of pulse grains. So just in closing, I'd like to acknowledge GRDC, Agriculture Victoria and University of Melbourne as well for supporting the image analysis research. Thank you.